This is the model answer video for option A, immunology and disease, and this is from your key assessment 1 this year. We've got some information about Lyme disease, and this is caused by a bacterium. Say bacterium B. Burgdorferi, and it's endemic. It tells us some other interesting information here. There are a number of strains of this bacterium. We're going to use that information later on. And it tells you a little bit about how you get infected and a little bit more information there. To start off with, an KA1, just give us the meaning of the term endemic, so there are no excuses for not getting this right. So it's a disease that that is always present in an area. So it's always present. So for example, in this country, apart from Lyme disease, things like the common cold or flu, it's always there. Someone's always got it, so it's a endemic. Other things like epidemic, which is like a major outbreak, or pandemic, across the world. But endemic is what we want. Label diagram to identify regions of an antibody. Again, 3 marks that you should definitely get. And A, we've got a bond between 2 polypeptide chains. So this is in the kind of tertiary structure. So this is a disulfide bond or bridge. B, they're trying to show you here where this can bind on something else. So this is the antigen binding site. So antigen binding site is where it's going to bind onto the part of the bacterium that it recognises. And C and D, well, C is just kind of a smaller version of D really. So C is the light polypeptide chain and D is the heavy chain. So light chain and heavy chain. So you should have got 4 marks so far. And indeed another 2 marks here, just AO1 kind of stuff, definitions and explanations. So an antigen. This will lead to the immune response and production of antibodies. So antigen will lead to an immune response, producing antibodies against it. So antigen, see, you can think of it as antibody generator. So it will lead to the generation of antibodies. And quite a few of you have just put that kind of 1 point there, but really we need 2 points here, 2 marks, where we explain the meaning of this complex. And it's not just, well, it's where they join together. We need a bit more information there. So the complex, so the antigen antibody complex, we need to say it's a specific antibody bound to the antigen, so, or it's the complementary fit. Specific's the best word though. So a specific antibody, so one that's been produced that's going to be complementary to that antigen. Specific antibody complementary. Let's say complementary. Complementary to the antigen binds to it. So I think you should have got 6 marks so far. We're now onto the body's response. So in the Help Me video, I coloured these lines in. For the primary response, that's in yellow here, the dotted line, and the after the primary response. And then you've actually got 2 different antibodies here, and we've got the second exposure here. So the secondary response is on the right hand side. So that's our primary response. So we've got a major peak from our dotted line in the yellow, and we've got a later peak from our blue line, our solid line. Then secondary response, a smaller response from the dotted line and a larger response from the blue line. Okay. So explain why the level of antibody G, so these are antibodies, immunoglobulin. It tells you up here 2 antibodies, and these are the names of them: IgM, IgG. I'm just going to call them M and G now. And explain why the level of G is higher after the second exposure. And this is due to memory cells. And we can give a further explanation there. So they make the immunoglobulin G during the secondary response, or after the second exposure, or due to clonal expansion, that sort of it. So make many immunoglobulin G after the second exposure. Lyme disease can persist in patients for many years, and the M remain high. Suggests possible reasons why. So M we get after, most of them after the first exposure. So this is where we're getting into not just regurgitating information now that we've had for the first kind of seven marks really. And it's given us some, if you're not sure about the answers to some questions, sometimes it's good to read the bump at the beginning of the question again. And it just gives us this little bit of information. There are a number of strains of this bacterium. So it's kind of leading us down to the fact that there might be different antigens, in the different strains, or even different antigens on the same strain, because we get this this peak in M after the first exposure. So we're thinking that the bacteria might have different antigens present on it, or antigenic variation. So bacterium may show antigenic variation, or different antigens. We could possibly suggest other kind of sensible suggestions that might produce this kind of first response. The evidence that we've got here, this produces a primary response each time, because we're saying the M levels remaining high. And we could say another possible suggestion, well, what about mutations? Well, if we have mutations of this bacterium, if it's still present, then that would produce these antibodies. Part D, we've got symptoms of Lyme disease. Include various different maladies there, and we've got a test against them. So what's happening with this test? We've produced a test well with, when we stuck onto this, some antigens. So we have put those on, and then we are exposing it to the these antibodies which are from the patient's blood. So this is from the patient's blood, but at the moment we can't actually see whether they've attached or not. Then we add, so this is from the patient, and then in the lab, so we've done this first one in the lab, and then we've added this from the patient, and then in the lab again we're adding these other confusing named antibodies, anti-human immunoglobulin G antibodies, but these are essentially just going to be complementary to the patient's antibodies, and just so that we will get this colour change if the patient's antibodies are there. So that's how that works. Using the graph, explain why the test would not detect the G antibodies in the days immediately after the tick bite. Well, the G in our blue here, in the days immediately after, there isn't any. So it takes 7 days for the G antibodies to appear. So immunoglobulin G not produced until day 7. And remember as well, we've got 2 marks here, so we need to say 2 things. I know we haven't got a lot of space. So why aren't these produced until day 7? Well, there might be a delay in the detection of the bacterial antigen, or latent period, or time for clonal selection. So do you know the story of how antibodies are made, and suggesting something along those steps that might take some time. So for example, delay in the detection of the bacterial antigen, so often called the latent period, or clonal selection, so whether the body is finding the right fit. So it's not produced, another way of saying it is time is needed to produce and secrete antibodies. So words to that effect. Part 2. The ELISA can produce false negative results for Lyme disease. So there's no colour change even if this patient is infected with the bacterium. So a range of answers here, and AO3 really, for 3 of these are kind of having to come up with some deep thought. So let's go with our first suggestion. Well, these antibodies here, well, why has the body produced these? The body has produced them to attach to the bacteria. So if they've attached to the bacteria, then they are not free to attach to our test antigens. So maybe the first obvious example is those antibodies are doing their job and might actually be attached to an antigen, so are not free. So antibodies are not free to attach to the test antigen as already attached to bacteria, or the bacterial antigen. You will notice here as well that we've got 5 marks, and so we're looking to come up with a point every kind of 2 lines or so. Quite a lot of answers that we've seen are taking up half the space with 1 point, and then half, the rest of it with another, and then coming right down the page. Really, we just want short, kind of snappy answers. Why might why might the patient be infected but this test doesn't show anything? Well, going right back to the beginning, there are a number of strains of this bacterium. So if it's a different strain, then these antibodies won't show up on the test. So they're different antibodies to the different strain. So the infection might be by a different strain of bacterium. So it mentions a different strain. So not recognised by the test. Then a similar sort of answer. Well, if this is a this is a bacterium, the antigen that we're using might be on the surface of this bacterium, or it might be a different antigen on the different part of the bacterium. So these antibodies might be concerning one of these antigen areas, whereas other antibodies will be produced against another antigen areas, and this is only testing for one of them. So there might be more than one type of antigen on the surface of the bacterium. So you're going to get different antibodies, of which this test is only really testing for one of them. Other suggestions, so we were kind of moving into the kind of experimental kind of faults. We're saying that the person is infected, but maybe they just aren't producing a lot of antibodies. So not many antibodies actually produced. So not many antibodies produced by the body. So no one's the same. You might produce a lot of them or not many. So there's variation in people's immune response. So variation in immune responses. Or indeed, this might make a colour change, but the colour change might be imperceptible. So another sort of way of saying this is only a small colour change, so you can't really detect it. Definitely going along the kind of experimental lines now. Well, we're kind of putting in a little bit of core information about enzymes and general things to do with experiments. So maybe the temperature that this test was done at wasn't any good for this enzyme. Maybe it got denatured or something like that. So maybe, for example, the enzyme's not functioning for some reason. There might be a pH problem, or something else is in the blood contaminating it. There might be some inhibitors or something like that. So the enzyme not functioning, and there's a range of things you could possibly say there, e.g. denatured. Because quite often these tests will have to be done under specific conditions, and maybe that hasn't happened. And we could make another suggestion for another control variable, for example pH, or not enough blood sample used, or just trying to think of other reasons why it wouldn't work. So for example, pH not right temperature. Well, we said denature. The temperature might be tested at too low a temperature, so we're not getting enzyme activity. So pH not right, temperature not right. Not enough sample. Those sorts of suggestions there. So quite difficult to get, to get 5 there. Most people could come up with possibly 3. And then the last part, hopefully a little bit easier. So we can test this bacterium by growing a bacterial lawn and use the discs. So you did this practical, and you can see the areas where the growth is inhibited. State the temperature at which the culture should be indicated. Well, it's 37 plus or minus a little bit, but 37 degrees C, as this is body. That's body temperature. It is a human pathogen. So this bacterium is going to grow. We're trying to grow this bacterium, and the best optimum probably for this bacterium is the body's temperature, as that's its normal habitat. Suggestions of 25 degrees C, because we don't want to grow pathogenic bacteria. Well, this is in the real world now, so we need to grow this as optimum. We're not really concerned about safety, trying not to grow human pathogens, because this is a human pathogen, so we are trying to grow it. So well done for thinking of 25 and remembering it, but in this case it's almost exactly the opposite. The most effective antibiotic was found to be E, and the zone of inhibition was the diameter. Diameter of the zone of inhibition was 22. Calculate the area. So handily gives you a formula for the area of a circle and pi, and we got the diameter equals 22 millimeters. That means the radius is 11 millimeters. So using our formula here, so the area equals pi r squared equals 3.14 times by 11 times by 11 equals 3.14 times by 11 times by 11, 379.94. And it does say to a suitable level of precision. So how do I choose a suitable level of precision? I look at the information that I've got. We've got 2 significant figures with my 22, and I've got 3 significant figures with my pi. So 379, well, I think we're happy there with 380, so I'm going to round that up to my 2 significant figures for my measurement there. Pi could be in any, couldn't it? So 2 significant figures for my 22 millimeters, and this is going to be 380. What units have we got? This is millimeters, and it's area squared. So we had quite a few just without the units, just with millimeters, or actually we had some millimeter cubed. So it's just math skills, and marks are hard to come by with this. Never mind throwing any away by being clumsy with the units. And being with reference to the image, suggest what assumptions being made when making this calculation. Well, looking at the image, we can see that the shape of this is actually something like that, but we're calculating a circle. So we're assuming that this is a circle for calculation purposes. So the zone of inhibition is a perfect circle. Or the radius is the same all the way around. Clearly it actually isn't in the picture, but that's the assumption we're making. Other possible assumptions that you might be thinking of is, well, I'm assuming that the clear area has got no bacterial growth in, but that's fairly obvious. And that's option A.